It is worth starting with the science here because it is, without exaggeration, one of the most conceptually elegant developments in modern medicine. The mRNA technology that produced COVID-19 vaccines in less than a year, itself an extraordinary achievement, turns out to be a platform capable of something far more ambitious. Tumors, it is now well established, accumulate genetic mutations as they develop. Those mutations produce unique proteins called neoantigens that are essentially foreign to the body's immune system. A personalized cancer vaccine does the following: sequence the tumor, identify its unique neoantigen fingerprint, manufacture a customized mRNA sequence that teaches the immune system to recognize those specific markers, and then deploy the patient's own immune system to hunt down any cell displaying that fingerprint. The elegance of this approach is that it is theoretically infinitely customizable. Every tumor is different. Every vaccine is therefore different. What Moderna and BioNTech have demonstrated in late-stage melanoma trials, significantly reduced recurrence when combined with checkpoint immunotherapy, is proof of concept at the clinical scale. These are not preliminary mouse model results. These are human trials in late stages with measurable outcomes. The therapeutic application, treating existing cancer by programming immune destruction of tumor cells, is the nearer-term reality. The preventive application, vaccinating high-risk individuals against cancers their genetics or exposures suggest they are predisposed to, is the longer-term and potentially more transformative horizon. Imagine BRCA mutation carriers receiving a preventive vaccine targeting breast and ovarian cancer neoantigens before a tumor ever forms. That is not an implausible 30-year vision. It is a plausible 15-year one. There are genuine scientific challenges that deserve mention. Not all tumors have high neoantigen loads. Pancreatic cancer and some brain tumors have relatively cold immunological profiles that make vaccine approaches less immediately effective. The technology will not work equally well across all cancer types, and the field risks overpromising if early melanoma and lung cancer successes are projected universally. But the equity question is the one that demands the sharpest attention. Bespoke cancer vaccines, manufactured individually for each patient based on their tumor's unique genomic profile, will initially cost hundreds of thousands of dollars per patient. This is not a rhetorical concern about fairness. This is a structural reality that will determine which populations benefit from this revolution and which do not. If personalized cancer vaccines follow the trajectory of other precision oncology drugs, they will be available in top-tier academic medical centers for insured or wealthy patients for a decade before any meaningful access expansion occurs. The science is ready to begin writing one of medicine's most remarkable chapters. The societal machinery required to deliver that science equitably is not. That gap is not a scientific problem. It is a political and economic problem, and it will not solve itself.